Okay, welcome student to the next topic. After completion of six kingdom classification, we are here for kingdom Monera in the biological classification. So let's begin. Okay, so whenever we see kingdom Monera, it is a exclusively kingdom of bacteria. It is exclusive. The sole members of this kingdom are bacteria. Okay, kingdom Monera. Bacteria, soul means only. Okay, so only member of this kingdom are bacteria. Again, these are the most abundant. Bacteria are most abundant. They almost occur everywhere, omnipresent, almost omnipresent. Okay, they will use such synonyms in the questions, and they also occur not only normal habitats where life is easily possible, but also in extreme habitat where generally life does not thrive. So we can take this example of hot spring. Hot springs are generally based on volcanic beds. Okay, the water moves through it, and these hot springs are also containing more amount of sulfuric acid. Hot thermoacidophils will survive here more. Okay, the water is acidic, also hot, scalding hot. Next is desert. Everybody knows surviving in a desert is very difficult due to the heat and amount of water and lesser biomass. Snow presents its own challenges. Okay, too much too low temperature and too much temperature pose a different thing. Here metabolic rates go down, not much of a biomass is there, and protecting from the cold itself is a challenge, but still bacteria survive here. And then we have deep oceans where pressure and temperature, all these things change. Those are not inhabitable. Only few extremophiles such as such bacteria will survive here. Another one is then on and inorganism. On means ectoparasites and in means endoparasites. The bacteria can be ectoparasite and endoparasites. They can live in the gut, on skin, okay, on various organs. So this is also parasitic mode shown by bacteria. So bacteria are grouped under this one is asked. They make question out of this based on their shape, and here they are going to make a match the matrix or match the pair question here. Next, pay attention again. Spherical coccus, singular is coccus, plural is cocci. Rod shaped bacillus and bacilli. Comma shaped is vibrio, that is vibrio, vibrium, vibrio, and spiral, that is spirillum and spirilla. You can see here. So spherical, bacilli, spirilla, and vibrio. Now pay attention to each and every structure. So single is monococcus, diplococcus. If it is a strip, it is called a streptococcus. These wordings will come later. So here you can see bacillus. Generally bacillus is a rod shape, and here whatever you are seeing at one end, the circular body, it is called as spore. Whenever bacteria is not having enough food or unfavorable condition, it goes into spore formation. So this is like very low metabolism to avoid the external unfavorable conditions. So this is spore. This entire structure looks like a spoon. Okay. Next we have spirilla. These are the flagella at present at the end. Okay, single one is called as flagellum, many are called as flagella, and this is like a spiral. So it is called as spirilla. Next we have vibrio. Vibrio is like a comma, so it is called as comma shaped. Simple. So this is the introduction of kingdom Monera, first kingdom, and here we have a very good question. Though bacterial structure is very simple, bacteria are very complex in their behavior. So, this is the statement given in NCERT, but there is no explanation how their structure is very simple. This could be understood because cell wall, cell membrane, glycocalyx, internal cytoplasm, and all those things. Okay, it is simple, but how their bacterial behavior is complex is not mentioned in NCERT. So one of the concept out of NCERT is the quorum sensing. Quorum sensing means this group sensing, simple word. These groups can sense if any threat is coming, such as antibiotic or any drug, so they form a film around them to protect it. This is called as a biofilm. Biofilm is becoming major problem for antibiotics. So they can understand this. So their behavior is not very simple. It is very complex. They can coordinate and express the genes for biofilm. So this is one of the examples. That is biofilm formation. And again, when one bacteria is resistant to some drug, it can share its plasmid to another which was earlier susceptible. So the plasmid sharing, drug resistance sharing, formation of biofilm, all these are complex behavior shown by bacteria. Next, bacteria as a group. Bacteria as a group shows most extensive metabolic diversity. That means they have each and every types of metabolic diversity. If you go for autotroph, they have photosynthetic, which is common one, but chemosynthetic is not common one. So chemosynthetic bacteria, they are heterozygous, they have parasitic and saprophytic also. So sometimes the question is asked, which group shows most nutritional diversity. The nutritional diversity is the another wording for metabolic diversity. Okay, so we will go by one. First we will discuss the autotrophs who make their own food, their own energy. So photosynthetic is a very common one. We have cyanobacteria, and which are also called as cyanobacteria are also called as previously blue green algae. But again, we know that blue-green algae and cyanobacteria are not one, because though it is a name used in past, blue-green algae is a problematic word here. Algae cannot be prokaryotic. Algae is not a bacteria. So it's called as misnomer. But why still it is mentioned? Misnomer are a thing which are so popular, though they are wrong, but they are used still. Okay, so that is called as misnomer. For example, if you say Xerox in India, we say Xerox for photocopy, right? So the excavator we call it JCB. So these are the misnomers. Actually, those are brand names, not the equipment. And here, cyanobacteria is a prokaryote, not an eukaryote, so we cannot call it algae. Next one. So chemosynthetic one. When we are discussing with the chemosynthetic one, we have Nitrosomonas and Nitrosococcus, Nitrosocystis and Nitrobacter. These are all chemosynthetic bacteria. We will study their examples and their role. So we have got photosynthetic, we have got chemosynthetic. Next one, heterotrophs. So what is going with the heterotrophs? Now pay attention here, it is a parasitic one. Here it is parasitic one. So parasites generally depend on other living organism. They harm them. Their interaction is going to be, for parasite it is plus, for host it is negative. So plus minus. Saprophytes have different idea. They live on dead organic matter. So they don't harm others, but they are benefited. So it could be like this. Saprophyte. They are benefited, but others are not harmed because they are dead. Okay. So this one, This point is important. Bacteria as a group show most extensive metabolic diversity. Next one. Next one is Archaebacteria. So in bacteria, we have two types. One is Archaebacteria, earlier discussed, and Eubacteria. The major differences are there, but if you want to remember, Archaebacteria does not have peptidoglycan. True bacteria, Eubacteria have peptidoglycan. Okay. There are many differences, but this is a key difference you can understand. So Archaebacteria are called as ancient bacteria, and because they are ancient, they can survive in harsh habitats. Okay, there is a logic here that harsh habitat, earlier Earth when it was cooling down and forming, it was not a nice place. It was like a literal hell, boiling water, extreme temperature, continuous rain, lightning. Okay, so in that situation these bacteria survived. So these conditions are very less and at very rare places, so they can survive in the extremely salty area. Those are called as halophiles. Those are called as hallow means salt loving. So extremely salt, this is a match the pair question. 100% this is going to be a match the pair question. And if you study content like a question type, you realize what they are going to ask and how they are going to ask. Hot spring are nothing but thermoacidophils. The water is going to be hot, it will contain some acid, so it is nothing but thermoacidophil. Next is marshy areas. Marshy areas are waterlogged. Marshy areas are waterlogged, in which generally the contents of plants are uh, mixed with soil, and there is the oxygen. Okay, so there methanogens produce most of the methane. Methanogens produce most of the methane. Next we have gut of ruminant. Gut of ruminant. Here ruminants such as cows, there they have two stomachs. One is ruminating chamber and another one is normal chamber. There the bacteria which has an enzyme that is called as cellulase, they break down cellulose. By a further process they generate methane. Methane is released by these cows. And again gut of termites, so this is not part of what syllabus. So again we have what ruminants, methanogens, and methane production. Okay, so simply these four match the pairs 1, 2, 3, and this one is fourth. This is going to be a match the pair question. Remember it. Next. So Archaebacteria, we can go with the Archaebacteria cell wall. Here, if you see peptidoglycan is absent. Peptidoglycan is low. Yes. Archaebacteria, if you can see here, cell wall, the peptidoglycan is absent. Peptidoglycan is a polymer of Nag and Nam, alternate polymer of Nag and Nam, which is present in Eubacteria. Okay, so in Archaeobacteria cell wall, peptidoglycan is absent, but some other proteins are present. These are called as S layer. Again, this is not part of NCRT, but good to know. These glycoproteins are present, which are responsible for survival of bacteria. Now see here, different cell wall structure is responsible for survival in extreme conditions. Different cell wall structure is responsible for survival in okay, it is not okay. So again, Archaebacteria having cell wall, peptidoglycan is absent, but S layer is present, glycoproteins are present, so that is there. Again, very key important thing here is different cell wall structure is responsible for survival in extreme conditions. They ask you question, what makes the Archaebacteria survive in extreme conditions? Is there different cell wall, nothing else, according to NCRT. Okay, but if you really see scientifically, their plasma membrane is also adopted. It's a monolayer which is also adopted as ether linkage between glycerol and fatty acid and branching of side chain Okay, branching of side chain. So this was our Archaebacteria. This was our Archaebacteria. Next we will start Eubacteria. Eubacteria. Okay, so now we are starting with the Eubacteria. Wait a minute. Let me take my mic. Okay, starting with the Eubacteria. Eubacteria is called as true bacteria. Eubacteria is called as true bacteria because it contains rigid cell wall where peptidoglycan is present. It contains rigid cell wall where peptidoglycan is present, and if motile flagellum will be present. If motile flagellum will be present. Okay, so let's get into it. Here, our first candidate of Eubacteria is Cyanobacteria. Eubacteria is okay. So let's begin with the Cyanobacteria. So Cyanobacteria are blue-green algae. Again, misnomer, but that is a famous name. They are photosynthetic autotrophs. They are what? Photosynthetic autotrophs. And what is mostly asked, I have seen a trick question. Cyanobacteria contain chlorophyll A. They contain chlorophyll A. Okay. So photosynthetic contain chlorophyll A. Again, they are present in unicellular form. That means an individual cell can exist, or they can be present in a colonial form. They can form colony, and they can be filamentous. Like this, they can form filament, spirogyre and all. These colonies are surrounded by gelatinous sheath. Okay. Now, again, if you go somewhere, it is called as also mucilaginous sheath. That is the difference between their monomer. These are different things, but can be considered one. Next. These are present in fresh water. These are present in marine, and also these are present on wet places in terrestrial. So fresh water, salt salt percentage is low. Marine water, salt percentage is high. In terrestrial, where there is a wetness, they will be present. Again, there is once environmental significance, blooms. Okay, the cyanobacteria do what? They form blooms. Suddenly their number increases. This happens when polluted water bodies, suddenly if some farm fertilizers run off due to what rain and accumulate in a pond or polluted water bodies, their numbers due to the nutrition they get, that doubles extremely. Their number increases overnight. That is called as water blooms. Okay, or algal blooms. So that is one of the thing. Next, we are going to fix the atmospheric. They fix atmospheric nitrogen in a very specialized cell called as heterocyst. We are going to see this in diagram. So the algae which fix atmospheric nitrogen is nostoc and anabaena. Nostoc and anabaena with heterocyst. A particular cell is there, and oscillatoria does it without heterocyst. So, but it all occurs in a rice paddy field. It all occurs in a rice paddy field. Okay. Next. So these are the characteristics of cyanobacteria. This way you can remember more. One more type of trick MCQ used to come earlier or maybe in DPPs. These are the examples of Cyanobacteria: Anabaena, Oscillatoria, Nostoc, Spirulina, Microcystis. Remember this name as Cyanobacteria. Okay. What happens? They will add this name between one of the option of normal green algae, and they will ask which of the following is a prokaryote or bacteria. Okay, so remember these names. These are cyanobacteria. These are cyanobacteria. Okay, so let's move on to the diagram. Okay, so when we are talking about the heterocyst, here we have filamentous blue-green algae. The example here is nostoc. So you can see here it has a heterocyst. It has what? Heterocyst. This heterocyst, again, this cell does not have chlorophyll, and this heterocyst, no oxygen, unoxygenic conditions are there, because they have to fix nitrogen. They have to fix nitrogen into ammonia like compounds, and this is going to be thick walled, so it does not permit any gaseous exchange and non-green. Again, there they called is gelatinous sheath, here they are calling mucilaginous sheath. Again, there can be a difference of monomer. And in nitrogen, one enzyme, key enzyme is there, that is nitrogenase. Okay. So for little bit concept clarity, we are going to go into depth of nitrogen fixation. This topic is not now, it is removed from syllabus, but just for the background information, we are going to go in depth for a little while. Okay. So this is nitrogen fixation. What happens? So heterocysts are where what is happening? Nitrogen fixation is happening. So again, this is a diatomic molecule of nitrogen, nitrogen, which is reduced to ammonia, This happens, nitrogen 8H plus 8 electrons, so 16 ATP, and that becomes ammonia, hydrogen 16 ADP, and all this. This catalyzed by nitrogenase enzyme. Okay, pretty simple. Why the nitrogen needs to be fixed? Because nitrogen is a valuable source in soil, and it is what all algae plants are competing with each other for. Here we have heterocyst and all other things. But okay, so here nitrogen, atmospheric nitrogen, it is reduced, and finally you can get the ammonia. They will not ask you this in this NEET. Just for background knowledge, you remember ammonia, nitrogen. It's converted into ammonia by nitrogenase enzyme. Then this enzyme, this reaction is reduction enzyme. So oxygen interferes with this. That's why there is no photosynthesis. Photosynthesis releases oxygen, and it is a non-green heterocyst. So it will reduce atmospheric nitrogen to ammonia. Okay, done. So we have completed cyanobacteria. Next. Next we have is chemosynthetic autotrophs. Which oxidize inorganic substances and get their energy. These are the chemosynthetic autotrophs. These that does not directly depend upon sunlight. Again, their ecological role is very important. They play great role in recycling nutrients like phosphorus, phos sorry, phosphorus, iron, nitrogen, and sulfur. Okay, so these are very important for ecological recyclation. Okay, If all the nitrogen, iron or nitrogen or sulfur or phosphorus become fixed, it will be unusable for other organisms. So these organisms oxidize them, release them into nature. So generally will be ammonia to nitrite, nitrite to nitrate. So these are nitrifying bacteria. One example we are going to take. This is also not present in NCERT, but science sake. Okay, ammonium is there, which is oxidized. Ammonium is there. Again, atmospherically they are going to fix. They are going to fix in cyanobacteria. These are going to release ammonium plus oxygen, nitrosomonas, nitrosococcus makes what? Nitrite. Okay, you don't have to remember this reaction, just study this. Ammonium is oxidized into nitrite and nitrite will be oxidized into nitrate. Okay, so this is very simple. Remember my NO2 trite, NO3 trait, just like that. Okay, so this is extra, not part of syllabus, but just to understand how this chemosynthetic bacteria works, study this reaction. Okay, next we have is a heterotrophic bacteria. So what is happening in heterotrophic bacteria? These depend on others, as they mention, for nutrition. They are not autotrophic like chemo and photo, and these are most abundant in nature. So majority of the bacteria, all bacteria, if you consider, this one is very beautiful here. Pay attention. If you consider all bacteria, 100% of them, majority are going to be what? Heterotrophs. So majority of heterotrophs are, again, out of them, majority are decomposer or saprophytic. Very important. Out of all bacteria, heterotrophs are more. Out of all heterotrophs, decomposers and saprophytes are more comparatively to parasites. Okay, next. So we are going to see first beneficial bacteria. Okay, so which ones are beneficial for humans, which ones are beneficial, harmful for humans again. So making curd from milk, basic concept. Lactobacillus converts curd into milk. It produces lactic acid. Sorry, milk into curd. It produces what? Lactic acid. Lactic acid curdles the milk proteins and creates curd. Fixing nitrogen in legume roots. Fixing nitrogen in legume roots. The nitrogen fixation here is done, and this is not a cyanobacteria, this is a rhizobium, and this is a symbiotic association in which both are benefited. Okay, in legume roots, what happens? They form a nodule. So plant provides food and ATP and all, and the bacteria fixes the atmospheric nitrogen into ammonia or ammonium salt. And finally, production of antibiotics. Okay, what is an antibiotic? A product, a secretion by one organism which stops or kills the other organism. It is called as, stops the growth of organism and kills the other organism, is called as antibiotics. Okay, so some of the examples, these examples you need not to remember, just for the sake of knowledge. We use streptomycin. Its source is Streptomyces griseus. Terramycin or oxytetracycline, again racemimus, erythromycin. These are one of the streptomycin, erythromycin. Okay, these are regularly used antibiotics which are the source, which has a source of bacteria. So these are beneficial ones for humans. Again, harmful. So harmful, which cause bodily damage or economic damage to human interest. So first here is going to be pathogen. Okay, pathogen is a damage to human being, crops, farm animal and pets, anything living related to human. These produce diseases and disorders. And then cholera, this is a match the pair. Okay, this is one of the match the pair that will come. They ask this often. Very simple. These chapters are very simple. These are data heavy. If you consider, remember and know few concepts, and most of the data, if you remember, it is so easy to get mm, all the questions right here. Okay, so cholera, Vibrio cholerae. Typhoid is Salmonella typhi. Okay, tetanus is Haemophilus tetani, and citrus canker is Xanthomonas citri. So what they will do, generally they will think that cholera to cholerae, typhoid to typhi, tetanus to tetani, citrus to citri. It is very easy to student to make this connection. So what they will do, they will not put these names, species name in the match the pair. They will put this, just these names, genus name, and it will make you match the pair. So pay attention, remember the whole name. It is so useful. Next. So we have completed heterotrophic, we have completed autotrophic bacteria. In autotrophic bacteria, we have completed cyanobacteria. Then cyanobacteria after, we have gone with the chemotrophic bacteria. Then heterotrophic bacteria, both beneficial and harmful. And finally, one that is remaining, which is also harmful, is a mycoplasma. Is a mycoplasma. Now pay attention. Mycoplasma comes under this. Mycoplasma comes under Eubacteria. Mycoplasma comes under Eubacteria, which is also called as PPLO, pleuropneumonia-like organism. Okay, so pleuropneumonia-like organism. Why its shape? Pleuro means many shape. Why it can show many shape? It is the Eubacteria which does not have cell wall. Now pay attention. It is important. It is a Eubacteria which does not have cell wall. Okay. It is the smallest living cell. It is the smallest living cell. Next, it can survive without oxygen. Can survive means it's not an anaerobe. It can survive without oxygen. So we can call this facultative anaerobe. Facultative anaerobe. Originally, it is a aerobic bacteria, but if it is not there, it won't die. It will survive by becoming facultative anaerobe. It is both pathogenic in plants and animals. Facultative anaerobe concept. Original aerobic. Okay. Originally aerobic, but can survive in anaerobic condition. This is the diagram. So you can see there is no cell wall. No cell wall. Okay. The next topic is bacterial reproduction. Next topic is bacterial reproduction. Okay. So here one MCQ, and generally what they will give you this here also. Pay attention how the MCQ is asked. They will give you these characters and will ask which of these character match to the mycoplasma. So simply can survive, there are keywords: can survive without oxygen, smallest living cell, and lacks cell wall. If these three are there, it is going to be mycoplasma. Okay, it makes the question solving very fast if you go by keywords. So again, in the bacterial reproduction, what they ask is binary fission. Binary fission is the main way of reproduction in bacteria, that is mainly by fissions. Bacteria grows its cell size, then divides into two cells. Pretty simple. It is called as binary fission. Binary means two, fission means breaking down. Okay. Then spore formation. This can be debated. Is it a reproduction or a survival mechanism? Because here cell number is not doubling. Okay. We are going to see all the diagrams there. Let's see binary fission. Okay. This is a binary fission. One bacteria was earlier, it grew into larger bacteria. Now it is making two bacteria, and it's in the end it will separate into two. Okay. So spore formation under unfavorable condition. You can see this diagram. In this diagram, the bacteria is forming a spore, which is called as endospore. The bacteria becomes spore, then it releases new bacteria. So there is not new bacteria formed, so it cannot be a reproduction, but survival mechanism. But NCERT says it's a reproduction, so reproduction. So it can be a source of perennation, survival. It is a source of perennation, survival. Next, the key is here is sort of sexual reproduction. Sort of sexual reproduction, primitive type of DNA transfer from one bacterium to another. Sexual is conjugation. This term sort of sexual reproduction is used by NCERT. Okay, whenever they say sort of sexual reproduction, you immediately jump to conjugation. Okay, so this is nothing but a primitive type of DNA transfer. Why it is called a sort of sexual reproduction? Generally, sexual reproduction means it brings variety. So what variety? Different. If a bacteria is sharing its plasmid with another, it is bringing different DNA information, and it is responsible for drug resistance. Okay, we can see it here. So this is a bacteria which has a plasmid. Okay, this is a bacteria which has a plasmid, and this one is does not. Those who have plasmid will have a pilus. That's why in early they will call it male. They will call it male, but not now. Okay, so female. So what happens is its tube is developed, that is pilus, and this plasmid is replicated and sent. And this plasmid contains antibiotic resistance gene. Earlier, this bacteria would die if exposed to, say, streptomycin. But now, if streptomycin resistant gene is transferred here, now both the plasmid have become what? Resistance. So, okay. So let's take a diagram of our own. Okay. So simply, this bacteria is sensitive to streptomycin, and this bacteria is resistant to streptomycin. Okay. So what is happening is this bacteria has this plasmid on which the resistance gene is there in red. What happens if I bring this streptomycin? It will kill it, but it will not kill the resistant. So what does resistance one do? It develops a pilus. It develops a pilus and enters the cell of the sensitive bacteria. And if you can see here, okay, okay, this what do you call this? Plasmid is now with the antibiotic resistance uh, gene attached to plasmid will be transferred through this into this bacteria. Okay, so when it is transferred into this bacteria, now this bacteria from sensitive it becomes resistance. So again, it cannot be now killed by streptomycin, so it is happier. So this is called as, and this is the mechanism of what? Drug resistance. This is a mechanism of what? Drug resistance by conjugation. And conjugation is nothing but sort of sexual reproduction. Sort of, not exactly. Because in exact sexual reproduction, there is a formation of gamete and all. But why we are calling it sort of? Because it is bringing in variation. And variation is the one of the characteristic of sexual reproduction. So here we have completed kingdom Monera. Kingdom Monera complete. So see you in next lecture. which we are going to discuss kingdom Protista. Thank you for listening patiently. Okay, go through it. I don't edit my videos much because again editing means it's like a classroom lecture. I'm not going to edit and ahas and all that blank space. If you go on editing, it does not create a flow. And so without editing, as it is the natural lectures. Okay, so see you in next lecture. Okay, we have completed kingdom Monera, and then we will see the Protista again is a eukaryote unicellular kingdom. Thank you. Keep watching the video and keep sharing.